the effects of changes in a single species may extend well beyond to the ecosystem. Single impacts are always joined by the risks of ecosystem damage and destruction. So here what happens, whatever the genetically modified organisms or the plants which we introduced into the ecosystem, it will damage the ecosystem or it will destroy the ecosystem. How it is going to have the negative impact on the ecosystem means, as in the previous slide already you have studied, whatever the GM crops which we have introduced, it is going to compete with the natural species for the available nutrients. So whenever it competes with the natural species, it will utilize the nutrients that are present in the soil and it will start growing very luxuriantly in the ecosystem, and it is going to replace all other varieties of the species or the natural species or the endemic species that are present in the ecosystem. Because of this, after some time, when you look into the ecosystem which was very diverse earlier before introducing the genetically modified crop, now you can find only in the nature or in an ecosystem only one variety of the plant, and that too, that variety is nothing but the genetically modified crop which we have introduced, and such condition is called as monoculture. Monoculture means only growing a particular type of variety or particular species in a particular area is called as what? Monoculture. Mono, single, culture means growth or culturing. So whenever we introduced the new genetically modified crop, what happens? It is going to overcome or it will compete with the species that are present in the ecosystem, and they will show invasive growth, and they are going to replace the other species that are present in the ecosystem, because of which we may lose the diversity that is present in the ecosystem. That is why in the slide you can find the genetically modified plants are going to decrease the biodiversity, and that will lead to what? Monoculture. Monoculture means only you can find in the nature or in the ecosystem only the GM crop which we are introduced in the field. So this is what the ecosystem impact. So in a single point you can find that it is going to destroy or it is going to cause damage to the ecosystem by decreasing the biodiversity, and thus causes ecological imbalance in the ecosystem. In the next slide, that is the fifth effect of the GM crops is adverse effect on the health of the people or on the environment. So first aspect you are studying allergens. Whatever the genetically modified crops or genetically engineered plants are there, they are found to be allergic for the people, or they are going to have the adverse effect on the human being. One example which I have given in this slide is inserting a gene from a nut into another plant could be dangerous for people who are allergic to nuts. Nuts means, here I can take the example of peanut. Peanut means commonly we call it as what? The groundnut which we use. Some of the people are allergic to the peanut. Some people, whenever they eat the peanut, they are going to show some allergic reactions like skin reactions, redness, swelling, itching, tingling, diarrhea, stomach cramps, nausea, vomiting. These all are the allergies that are shown by the people which are allergic to peanut. Sometimes what happens, the genes are isolated from the pea plant and then they are transferred into the other plants. If the people which are allergic to peanut, they consume the genetically modified crop which contains the genes from the peanut, are able to show some of the allergic reactions to the genetically modified food. So this is one aspect, that is allergy is also caused on the human health. The second aspect which we need to study is antibiotic resistance. This we have already studied in the horizontal transfer of the genes. So here one thing which I can say, usually while developing the genetically modified crops through genetic recombination or through genetic engineering, the scientists or the bioengineers use the marker genes. Usually the marker genes used are the antibiotic resistant gene for the selection of the transformants and the non-transformants. Transformants means, always remember, cells which have received our desired gene, or the plants which have our desired genes are said to be transformants, and the one which have not received the genes are said to be the non-transformants. So in order to identify the transformants from non-transformants, what the bioengineers do, they will insert the marker gene. If these marker genes are introduced into the food chain, so already you know, through horizontal gene transfer, that is from the genetically modified crop into any microorganism, if these antibiotic resistant gene, you leave about the direct transfer. Direct transfer means directly from the genetically modified organism into any bacteria that is present in the soil, that is present in the nature, you call it as what the direct transfer. Sometimes what happens, whenever these genes are going to enter into the food chain, and if they are taken up by the human gut microflora, human gut microflora means usually in the digestive system of the human gut, the bacteria that is present is the Escherichia coli or the E. coli. E. coli in the human gut that is present is said to be the non-pathogenic. So sometimes what happens, due to the receiving these antibiotic resistant genes from the genetically modified crop, what happens, the non-pathogenic E. coli bacteria may become harmful disease causing bacteria, and it may have very bad effect on the human being. So this is what the adverse effect on the health of the people and the environment. And the sixth one, the sixth bad effect which we need to study is, new harmful bacteria and viruses may be used as a warfare agents and can cause havoc. So the same thing here, through recombinant DNA technology or through biotechnology, what the bioengineers are doing, they are developing many bacteria and the viruses which are harmful or which can cause the diseases. So daily in the laboratory, many researchers are going on the bacteria and all, but because of some issues between the countries and all, sometimes what happens, the bioengineers may develop such a bacteria or such a viruses through biotechnology or through genetic engineering, and we call those bacteria or the viruses as what, the genetically modified organisms which have the pathogenic or the virulent genes that are present in them, and those can be introduced into the environment and which can cause the diseases. So already the now era or nowadays, whatever the issue that is going is COVID-19 disease, which is caused by the coronavirus. Some of the scientists or some of the other American country is telling that whatever the coronavirus is there, it is developed by the China. So what they say is, purposefully China has developed this and it has used this as a biowar, but we don't have any clues to blame the China. What they say is that coronavirus is developed by the bioengineers of the China, and they have released that into the environment in order to make the world to suffer, or in order to become king in the global world, it is said, but we don't have any proof to blame the China. So like this, in the laboratory, many harmful viruses, bacteria which can cause the diseases can be developed through genetic engineering, and we call them as what, the genetically modified organisms, and are able to cause the disease. This is what we are studying in this slide. And in the next step, any accidental escape from the laboratory strains may be used as a warfare agents and can cause havoc. In the previous slide, we studied that purposefully the bacteria and the viruses are developed in order to create the havoc. But sometimes what happens, due to the carelessness of the research fellow who is working in the laboratory, or by the accidental escape from the laboratory, say for example, some are working on the HIV virus, and sometimes it is also said that whatever the coronavirus is there, it is leaked from the Wuhan laboratory when they were working on the virus, that is HIV virus, or some say that that is when they are working on the drug development against the HIV, the RNA material of the virus is being mutated or modified and it is accidentally escaped from the laboratory, and now it is able to cause, or now it has become the pandemic disease all over the world. So because of all these reasons, sometimes accidentally what happens, the genetically modified organisms, if they get escaped from the laboratory, means they may cause any kind of the bad effect on the environment, and that could be used as warfare agents and can cause the havoc. And the next or the last topic which we are studying, or the last threats or the risk of the GM plants, is that GM plants and animals may create sociological and ethical problems. We know very well ethics means person's beliefs and the values. So whatever the ethics is there, it believes on the food security and the effects on the health. We know how the genetically modified crops have the effects on the health, that is they may cause the allergy. This is all. And security of the food is whenever we introduce them, you know very well, horizontal transfer of the genes, which may increases the risk of developing the diseases in the human body and all. These all are what comes under the ethical problems. And sociological and ethical problem means some of the person's belief and the values, what they think that we are going against the nature, we are going against the God's creation, it may have some bad effect on the health. We are not supposed to eat the genetically modified crops. If not today, in future, say for example after 10 or 20 years, they may have the bad effect or the adverse effect on the human being. Thinking this all things, the social and ethical problems are hindering the introduction or the research in the development of the genetically modified plants. So these all are what the risks and the threats of the genetically modified plants. Thank you.